Welcome and thank you for attending our Ahead of Cancer lecture series this evening, sponsored by the Allegheny Health Network Cancer Institute. My name is Margie Leslie. I'm a director for the Cancer Institute. There are a few housekeeping items that we want to review before we start. During the presentation, all participants can type questions into the chat or the question and answer area portion of the Zoom. We will be addressing those questions at the end of the lecture if time allows. Participants are not able to speak during the conference, but again, we encourage feedback via the chat option. Tonight, we're excited to welcome Drs. Lister, Samhouri, and Donnenberg, who will be speaking about harnessing the immune system to fight cancer, from vaccines to cellular therapeutics. Dr. Lister is the chief of the Division of Hematology and Cellular Therapy at Allegheny Health Network Cancer Institute. He is an experienced transplant physician who participated in his first bone marrow transplant in 1979. Since that time, he has been involved in more than 1,000 transplants and has contributed to many clinical studies and made original contributions to the literature. Dr. Lister received his initial exposure to marrow transplantation as a trainee at McGill University and received further training at the Frederick Hutchinson Cancer Research Center in Seattle. Subsequently, as one of the first 4 Terry Fox Fellows in the National Cancer Institute of Canada, he received additional clinical and laboratory training in marrow transplantation at the Ontario Cancer Institute, Princess Margaret Hospital at the University of Toronto. He returned to McGill and actively participated in transplant for the next 5 years before accepting a position in the Division of Hematology and Bone Marrow Transplantation at the University of Pittsburgh in early 1992. In 1997, he joined the faculty at West Penn Hospital. Dr. Lister is a professor of medicine, Drexel University College of Medicine, and serves on numerous scientific administrative committees, including the Lymphoma Committee of the CIBMTR. He lectures internationally on lymphoma and CAR T cell therapy. His research concerns the manufacture of CAR T cells and potential modifications to improve their efficacy. Next, Dr. Yazan Samhouri is a physician with the AHN Cancer Institute, specializing in hematology and cellular therapy. He is skilled at treating patients who have hematologic and lymphoid malignancies and various cancers of the blood. His expertise in therapeutic treatments using bone marrow transplantation and immunotherapy methods, including CAR T cell therapy and tumor-infiltrating lymphocytes. He received his medical degree from the University of Jordan in Amman, Jordan. Dr. Samhouri was chief medical resident at Rochester Regional Health, Unity Hospital in Rochester, New York. He completed his fellowship in hematology and medical oncology at Allegheny Health Network Cancer Institute in Pittsburgh, Pennsylvania, where he served as a chief fellow. Dr. Samhouri is certified by the American Board of Internal Medicine. He is a member of the American Society of Hematology and American Society of Transplantation and Cellular Therapy. Dr. Samhouri sees patients 18 years and older. Dr. Albert Donnenberg is the vice chair for translational research at Allegheny Health Network Cancer Institute and a professor of medicine at Drexel University College of Medicine. His current research interests are in cellular therapeutics, drivers of the epithelial to mesenchymal transition in neoplasia, and immunotherapy for metastatic cancer. Projects he pursues with these projects he pursues with his science and life partner, Dr. Vera Donnenberg. Dr. Donnenberg received his BA from the University of Colorado Boulder and his PhD in infectious disease epidemiology from Johns Hopkins, studying cellular immunity to herpes simplex virus. He spent many years at Johns Hopkins, where he became an associate professor of oncology. He worked on adoptive transfer of donor immunity following allogeneic bone marrow transplantation and pioneered the development and clinical implementation of bone marrow lymphoid depletion to prevent graft versus host disease. He also performed early studies on cellular immunity and HIV infection and co-developed the concept of T-cell hemostasis. Dr. Donnenberg joined the University of Pittsburgh to serve as the Director of Laboratory Research in the Division of Hematology and Bone Marrow Transplantation. He also served as BMT Program Co-Chair, Professor of Medicine, and Director of the UPMC Adult as well as Pediatric Hematopoietic Stem Cell Laboratory during his tenure. He is internationally recognized expert in cellular therapeutics, expert in therapeutic cell processing, on flow cytometry, and inspection team leader for the College of American Pathologists. Dr. Donnenberg has co-edited 2 editions of the CRC Handbook of Human Immunology and has authored more than 200 scholarly publications. It's my honor to welcome Drs. Lister, Samhouri, and Donnenberg to present this evening's lecture.
Thank you, Marjorie, for that introduction. My task to start out this evening is to give you information about chimeric antigen receptor, or as it's abbreviated, CAR T-cell therapy. So in this short lecture, I would like to answer for you several questions. The first is, what in fact is a chimeric antigen receptor? Then how do you get this chimeric antigen receptor into a T cell so that it can perform its effector function and kill cancer cells? And how, in fact, does it do that? I'll also talk a little bit about the commercially available products, just so you can hear their rather unpronounceable names. And we're going to speak a little bit about the major toxic events associated with this form of therapy. I'll end my presentation with a look at the research we're doing to try and to improve CAR T-cell therapy and how it might give our patients hope in the future. CAR T-cell therapy is only one type of immune effector cell therapy, which is abbreviated IEC. Another type, of course, is tumor-infiltrating lymphocytes, which are abbreviated TIL, which Dr. Samhouri and Donnenberg will describe in detail in their work. Let's begin by looking at what a chimeric antigen receptor looks like. So this is a cartoon. You can imagine a T-cell magnified many millions of times so that you see the membrane around which that the cell has. And on the outside, in the top portion, you will see that there is a molecule that spans the membrane and has pieces that are outside the cell and inside the cell. The piece that is outside the cell and labeled VH and VL, in fact, will bind to a cancer cell. This binding then transmits a signal internally to the cell through a co-stimulatory domain, and then ultimately through something called the CD3 zeta chain, which signals the T cell to begin to arm itself and perform its killer function. This is simply another way of looking at a car. If you look at the rectangles that are labeled 1563, this is what we are using in our own CAR T-cell program here at AHN. On the left, there is the binder, which is consistent with the prior cartoon showing the VL and V heavy, which is the portion that will bind to the cancer cell. There is a hinge region from a molecule, a normal molecule that T cells have called CD8, and the portion that goes through the cellular membrane is from another molecule that's normally found in the human genome. The co-stimulatory domain that we use is labeled 41BB or CD137, and the activating areas from CD3, a molecule which all T cells possess. So the reason that this is called a chimeric antigen receptor is that it sticks to something on the outside, and it's made up, much like Frankenstein's monster was, of a number of elements from the normal human genome. One of the things that is easy about this is that you can put something into solution and have it attached to the FMC63 portion of the receptor that glows with immunofluorescence. And when you look at that in the curves below, you can see that we have a an easy technique to recognize CAR T cells. So how do we make CAR T cells? Well, if we start at the top left, we will remove blood from a patient to get their T cells. Now, this process of removal is termed apheresis and is associated with a particular device that we use to process the blood. Once the cells are collected into a permeable plastic bag, we take them to the laboratory and insert them into a machine that I will describe later that will purify the T cells from the peripheral blood and allow us to insert a gene into the T cell that will then make the receptor that I described on the surface of the cell. The CAR T cell, thus, is a normal T cell that has this chimeric antigen receptor on its surface that allows it to eventually bind to cancer cells. We then use the device to grow these CAR T cells to millions of them and then infuse them into the patient where they wander around the body, and when they come into contact with a cancerous cell, they will become activated and perform their natural killer function. So this cartoon simply reiterates what I just said. On the left, you have a CAR T cell. On its surface, it has many molecules, but one of those is the chimeric antigen receptor. The chimeric antigen receptor can bind to an antigen on the surface of a cancer cell and release certain compounds into the cancer cell that will kill it. The tumor cell undergoes a process called apoptosis. So I'm having a little difficulty. So what then are the commercial products that are available here at AHN? They're listed here, and I only put them on the screen to show you that 4 of them are simply there for the treatment of B-cell lymphoid malignancy, such as B-cell lymphoma, CLL, and 2 have recently been approved for the treatment of multiple myeloma. And then, of course, we have our own internal product. I put this slide on simply to show you how nascent this field is. The first product was only approved on August 30th, 2017, and then was rapidly followed by other approvals, and there are more approvals coming for these products. So this is a very dynamic and rapidly changing field. Of course, like all therapy, this therapy is associated with toxicity. On this cartoon, you will see that the days are numbered 0, 7, 14, and 28. Day 0 is the day the patient actually receives the cells. During those first 14 days, there is the possibility, and with much less frequency even thereafter, the appearance of a toxic syndrome termed cytokine release syndrome, or CRS. We also recognize that there is another separate form of toxicity called the immune cell-associated neuropathic syndrome. And this tends to occur a little later. It can occur with CRS or without CRS. And we have clear ways of diagnosing these forms of toxicity and protocols to treat it. Usually this toxicity resolved within about 14 days. CRS can get very severe, and it can affect almost any organ system in your body, as detailed in this cartoon. Not only is there CNS, central nervous system toxicity, but it can affect your liver, your bone marrow, cardiovascular system, your lungs, your kidneys, your gastrointestinal tract, and even your muscles. Most of the time, though, we're able to avoid such severe toxicity by recognizing this toxicity early and treating it in a rapid fashion. Our own experience here at AHN involves the production of CAR T cells on clinical trial. And we use this device, which is self-contained and takes CAR T cells from an apheresis product, and at the end harvests the final product, which we can administer to patients. This clinical trial has been running, and we have 8 patients soon to be treated. I think we've demonstrated that this is feasible and that there is efficacy. This will be a confusing panel for you to look at, but I simply put it here to show you that we have an automated device in flow cytometry that allows us to identify CAR T cells and enumerate them in the product that we will administer to the patients. I would draw your attention to the very center panel in this cartoon, and you can see that there are 2 populations of cells, one on the left, which are cells that do not contain the CAR construct, and one on the right that demonstrates the cells that contain the CAR construct and are thus CAR T cells. So this is an automated procedure that we're able to perform rapidly to be able to dose the CAR T cells correctly for our patients, because this allows us to quantify them. As for the results, primarily the technique we use to show that the cancer has disappeared is a PET CT scan. And this is one of the patients on the trial. You can see on the leftmost panel that there are a number of white spots. I would point out that if you can recognize the anatomy, there is at the top the head with 2 arms sticking up and 2 legs at the bottom with the thorax and abdomen in between. A number of these white spots are actually normal excretion by the kidneys into the bladder, but on the left side, there are a number of white areas, and also on the right side, a number of white areas that represent lymphoma deposits. You can see that at 30 days after infusion of the product, almost all of the disease has in fact disappeared, and a repeat at day 100 shows clearly that the patient is in complete remission. This is just another way of looking at a PET CT scan where you seem to slice as though you're taking a slice of people and looking up from the feet. What you see here on the left cartoon, and then in more detail on the left, is the liver with some lymphomatous deposits, and on the right part of the left cartoon, white spots, which are in fact lymphomatous deposits in the spleen. 100 days after receiving CAR T cells, this patient was in complete remission also. So this is a complicated process here. It involves starting materials, which are the cells we collect from the patient, and the lentiviral vector that's used to transform the T cells, substances that are used in the manufacture and the final product, and also ancillary materials that we need for the culture of these selection of the T cells and the culture of them, and also their activation. So not only do we have these in clinical trial, we have conducted clinical trials with other experimental forms of CAR T, and I'll describe perhaps three other initiatives that we've had in our division over the last few years. So there was one we termed an allo CAR, or maybe a parked CAR, and this particular product is made from a single donor, and it is genetically modified in 2 particular ways. And also it's administered with a monoclonal antibody to get rid of the patient's T cells. So this could be an off-the-shelf CAR, something that could be produced and then used in clinical trial. It remains to be demonstrated that the efficacy of this type of product is identical or better than what is available commercially. To a CAR, you can also add other things that might make them more aggressive when they get into the body. And we had a trial using a synthetic form of a normal human molecule termed IL-15 that would perhaps increase the activity of the CAR T cells. And we also have the availability to provide a type of chimeric antigen receptor that targets more than one molecule on the surface of a malignant B cell, targeting both CD19 and CD20. So these are among several other entities that we've done. Of course, our local production continues to go on, and we believe that this is a way to rapidly provide patients with a living product that is not frozen before it's administered to the patient. So in conclusion, I think we're on the verge of a paradigm shift in the treatment of these types of cancers, and it will hopefully replace conventional chemotherapy, which often engenders longer-term side effects that negate the positive effects that the chemo might have. We believe the way forward is rigorous clinical investigation and laboratory research to better exploit the vulnerabilities we can identify in cancer cells. And I would be remiss if I did not thank all the patients who have participated in our clinical research and who altruistically will make things better for those generations to come. Thank you.
Okay, we'll move on with Dr. Samhouri's presentation.
Good evening, everyone, and thank you for being here to talk about cellular therapy and cancer in general. For my part, after listening to Dr. Lister talking about CAR-T for hematologic malignancies, I'll talk about cellular therapy for solid tumors and what are the 7 important things to know about that. I'll touch base about the recent approval of TIL therapy, which is the first cellular therapy approved for solid tumors in metastatic melanoma, which is lifileucel or Amtagvi. And then I'll talk about what available cellular therapies are at AHN. So the first thing is, what is cellular therapy? So lymphocytes or white blood cells are an important part of the immune system that helps the body to fight off infections or eliminate diseased cells. Lymphocytes, made up of T cells and B cells, are constantly patrolling the body to identify cells that shouldn't be present, including cancer cells. As cancers grow, those lymphocytes will recognize these cells as abnormal and penetrate into the tumor. Once they are in the tumor, they begin working to kill cancer cells. And this is the ideal situation, that if the immune system is 100% healthy and working, then we don't have cancer, right? But that's not the way. And why is that? Because those T lymphocytes and B lymphocytes are prevented from doing that by either breaks in the immune system or by signals from the tumor that weaken the immune system. And popular medications that came up in cancer medicine in the last decades, which are immune checkpoints inhibitors, and you might know them as Opdivo or Keytruda, were developed to block some of those breaks and unleash the immune cells to attack cancer. Now, what we are thinking about is how to use these T cells or lymphocytes with some improvement as a form of cell therapy to fight cancer, because we know their ability to penetrate these tumors, to recognize these tumors and kill cancer cells. How can utilize that knowledge and function of the immune system in our patients' advantage? The second thing to talk about today, what are the different types of cellular therapy? When you talk about cellular therapy, there are 3 types of cellular therapy. Dr. Lister already talked about one of them, which are CAR-T cell therapy. It's more in an advanced stage of development in hematologic malignancies, and we heard what Dr. Lister told us about lymphomas. It's not as developed in solid tumors yet. It's in the very early phases. But we have another cellular therapy called TCR cell therapy. and we have TIL cell therapy. The majority of the presentation, my presentation today will be about TIL cell therapy, but I'll mention quickly what are the differences between those three cell therapies. First difference is the source of the cells. Where are we taking these cells from? TIL therapy, it stands for tumor infiltrating lymphocytes. So those lymphocytes are taken or isolated from the tumor itself. So they are naturally occurring lymphocytes that are already in the tumor. We just take them and grow them in the lab. We don't genetically modify them. So those are naturally occurring cells. TCR and CAR-T, those two technologies are genetically modified. Why? Because we don't take them from the tumor, we take them from the peripheral blood. So those are normal T cells in the peripheral blood. We didn't take them from the tumor, we send them to the lab, we genetically modify them to teach them how to recognize the tumor. Tumor infiltrating lymphocytes, they already know how to recognize the tumor because we took them from there. Those are the core or major differences between the three cellular therapies. And the majority of the presentation now will be about TIL therapy because it's the most advanced stage in solid tumor compared with TCR and CAR T cell therapy. The third point to know about today, or the third question today, how can be these cells used for cancer therapy? Because TILs, as I mentioned, come directly from the tumor. They already recognize many targets on the cancer cells. This makes them a very attractive therapy because we don't have to do anything to point them toward the tumor. And these things that we do, as we do it in CAR-T and TCR cell therapy, just to teach them how to go to the tumor, we don't need to do that for TILs. A group of TILs taken from a patient's tumor may recognize many unique targets in the tumor, not only one target. And sometimes we don't even know what do they recognize on the tumor. But this offers a real therapeutic advantage because it prevents the tumor from evading our efforts by hiding one target at a time. Now, to use TILs as a therapy, we must help them overcome the hurdles in the tumor microenvironment and effectively eliminate the cancer. How do we do that? By two ways. First of all, expanding the TILs, because once we expand them in the lab, we can give the patient a much larger army of immune cells that is already trained to recognize and attack the patient's specific tumor. The second way we help the TILs to work better is by making the immune environment in the body more friendly to these TILs. And that's by giving a short course of chemotherapy before infusing the TILs, which I will talk about more in a minute. Now, the fourth question is, what's the treatment process for patients who undergo TIL therapy? So TIL therapy for non-melanoma solid tumor, and I specifically say non-melanoma solid tumors because we have one FDA-approved product for melanoma, but the rest of TIL therapy, it's still all investigational under clinical trials. For patients who enroll in a TIL clinical trial, the process begins with a tumor biopsy. So it's not really a needle biopsy. It has to be a large piece of the tumor to be taken out. And that part of the tumor will take to the clinical lab and then isolate TILs from that tumor. Then we do some expansion and sometimes engineering in a process that typically takes about a month for the cells to be ready. And one thing that we're working on to improve is how our speed in this area to get therapy to patients more rapidly. And Dr. Donnenberg will talk to us about some of this work that we are trying to do. Now, when the TILs are ready after this month in the lab, we give the patient a short term of chemotherapy regimen. And that chemotherapy regimen is not meant to treat the cancer. That chemotherapy regimen is meant to prepare the patient for the incoming cells. After we give the short-term chemotherapy, the patient receives the cell infusion, and those are the patient cells that we took from the tumor sent to the lab, expand in the lab, and we give them back to the patient. This is the treatment. This is the drug, actually, not the chemotherapy before, not the immunotherapy after that I will talk about. The drug is really the cell infusion. It's typically like a blood infusion. Once infused, the TILs travel directly to the tumor to begin their work by killing cancer cells. Now, after we give the cell infusion, the patient receives some immune modulating therapy, which is interleukin-2. The function or the purpose of interleukin-2 is to stimulate the TIL activity. So it's a stimulatory agent that we give the patients to help the TILs to grow and expand and kill the cancer cells better. It's important to know that TIL therapy, like CAR T cell therapy, is a one-time treatment. A patient will be in the hospital for at least 2 to 3 weeks so we can monitor for any side effects or reactions to the therapy. The fifth question for today is, what are the expected side effects to TIL therapy? So far, we have not seen any major side effects from the TILs themselves. And again, this is the drug, right? The cell infusion. But the majority of side effects of this sort of treatment, it's either coming from the first part of treatment, which is the short course of chemotherapy, or from IL-2. Toxicity from chemotherapy is like any traditional chemotherapy toxicity. Hair loss can happen, blood counts will go down, nausea, vomiting, risk of infections, like any other chemotherapy regimen. The IL-2 is an immunotherapy. So what it will do, it will stimulate the immune system. So the patient might have fevers, low blood pressure, and low oxygen, and needs really close monitoring during IL-2 administration. And because IL-2 can cause fevers and low blood pressure and low oxygen, one of the things that we're trying to improve in TIL therapy is how we can just eliminate the need of IL-2 by adding immune stimulatory signals that come with the cells themselves without adding an extra a drug like IL-2. And that will help make TIL therapy more logistically attractive. The sixth question for today is, has TIL therapy been successful in treating cancer? Now, we have been evaluating TIL therapies in clinical trials for years, and there are encouraging results in early phase studies. Most of the studies thus far have treated patients with melanoma, but other indications are now being explored. We have almost a TIL clinical trial for almost every cancer type. What is interesting, and that's really the important part about new immunotherapeutic approaches like TIL therapy, is the long-lasting response for these therapies. Chemotherapies, we developed several agents over the 90s and early 2000s, but chemotherapy will work. The problem with it will work for short term, and then the cancer can come back. The only therapies that can work for long term if you manipulate the immune system, either by using drugs like Opdivo and Keytruda, or by doing cellular therapy. And cellular therapy, it's even the best in doing so because there is the memory function of those immune cells or T cells that will keep patrolling the body. And once they recognize the same tumor years after, they will differentiate into a killer T cells and kill that tumor. And that's really the main advantage of cellular therapy as a novel therapy in this era. While this is still early data, and when I talk about early data, it applies for non-melanoma solid tumors because in a minute I will talk about advanced data actually for the first approved product for melanoma. We are still working very hard to understand how these long-lasting responses happens and how can we maximize the benefit for all these patients. We're also working on improving the manufacturing process. It's either by making stronger TILs or make it to finish the manufacturing process faster so we can make this therapy available to the patients quickly. Why are we excited about the future of TIL therapy? So we believe that TIL therapy offers a promising option for patients with solid tumors. So far, we have not seen great success from CAR T or other cell therapies in treating patients with solid tumors. On the opposite, CAR T is in very advanced stages in hematologic malignancies, as we learned from Dr. Lister tonight. However, by the very nature of TILs, we have years of data demonstrating them to be more successful for these cancer types. The FDA approved the first TIL therapy for metastatic melanoma, and this is the first cell therapy FDA approved for any solid tumor in the history of cancer medicine, actually. And that happened just in February 2024. And as Wayne Gretzky likes to say, skate to where the puck will be, not where it is. And we believe TIL therapy is where the puck is going. It's poised for a significant impact in the field of cell therapy, and we're proud to be part of bringing those advances to our patients. So what's the patient journey when they get TIL therapy? The first step is to get the tumor biopsy, or we call it a tumor procurement or harvest, which usually requires a surgery to take around 2 centimeters of tumor tissue from the easiest accessible site. And then we'll send this part of the tumor to the lab where TIL production will happen. This period, this step 2, it takes around 3 to 4 weeks to finish. So the patients will do surgery, will go home, will wait for 4 weeks. During those 4 weeks, he's allowed or she's allowed to receive bridging therapy, which is standard of care chemotherapy or immunotherapy, to get them to step 3, when we admit them to the hospital to start the 3 steps or 3 stages of treatment in the hospital. A short course of chemotherapy, cell infusion is the second step, IL-2 administration is the third step, and then recovery few days after the last IL-2 dose, and the patient goes home. And after that, no more treatment will be administered. We just repeat scans or repeat imaging and blood work to make sure the patient is responding to treatment. Now, this is lifileucel or Amtagvi, which is the first FDA-approved treatment after progression on checkpoints inhibitor, which we call them anti-PD-1 inhibitor, or the second therapy that is available for melanoma is BRAF and MEK inhibitor. Metastatic melanoma, there's almost 13,000 metastatic melanoma per year in the United States. 8,000 of these patients dies every year in the United States. 9,000 of these patients makes it to the first-line therapy. Second-line therapy, they drop to 6,000, and third or fourth-line therapy is almost 5,000. So you see, after first line of therapy, there's really no good treatment. And more than half of patients progress within 12 months on current first-line immune checkpoint inhibitor, which are Keytruda, ipilimumab, and nivolumab for melanoma. So we were really desperate for many, many years and decades, actually, about a therapy that helps us after patient fails first-line in melanoma. Amtagvi or lifileucel is preferred second-line or subsequent therapy in the NCCN guidelines, which is the main guidelines that we follow to guide treatment options for patient. This graph will show you the most interesting part in cellular therapy in general, which is these responses that last for years. I mentioned in the previous slide that more than half patients will progress within 12 months after failure of first-line therapy. So this graph before Amtagvi will drop like this. But with Amtagvi now, we can see this nice tail at the end of this curve. This tail means that more than half of the patients are still responding more than 4 years from TIL therapy. And this was unheard of in melanoma. What do we have at AHN? The 3 types of cell therapy that I mentioned, which are TIL, TCR cell therapy, and CAR T cell therapy. For that, I'm talking about solid tumors. So in addition to what we have for what Dr. Lister mentioned, we have almost 10 clinical trials for TIL therapy that covers almost the majority of cancer types, which is lung, colon, melanoma, breast, ovarian, cervical, endometrial. We have 1 clinical trial looking at TCR cell therapy that covers lung, head and neck, melanoma, cervical, and ovarian. And we have one CAR T cell therapy for solid tumor that is coming soon, that is starting for gynecologic malignancies, but it will open colorectal. It will open for patients with colorectal cancers, breast cancer, and lung cancers. That's all what I have for today, and I'll be happy to take questions at the end of the webinar. Thank you.
Okay, we will transition to Dr. Donnenberg. Dr. Donnenberg?
Good evening. So today I'm going to talk about our future directions, particularly in-house manufacture of biological therapeutics. And I will start with a virtual tour of our new facility for the manufacture of cellular therapeutics. And I'll tell you a little bit about fast TILs. We call them fast TILs because we can make them in 8 days rather than the month that it takes to make TILs usually. And local delivery to treat cancers metastatic to the pleura. I'll also talk a little bit about in-house manufacture of conventional and tumor-targeted TILs and of immunologic priming of tumors with oncolytic virus prior to in-house manufacture of TILs. Oncolytic virus, onco, cancer, lytic, meaning bursting. So oncolytic viruses are viruses that are designed to kill cancer cells. So we'll start with a virtual tour of the new facility. This is at West Penn Hospital, and it was one of the oldest parts of the hospital and one of the most dilapidated. And you can see that after this massive $12 million effort, we have state-of-the-art laboratories. So I'm going to give you a virtual tour. Well, this is a blueprint. And what you can see is that when you enter into a research laboratory. And this is where we do preclinical experiments to develop and test new ideas. This is the clinical testing laboratory. And this laboratory is used to perform quality control and other measurements on cellular products that we are actually giving to patients. And then we have 2 clean rooms. One is for CAR T and TIL production, and the other is for the production of oncolytic viruses, and also to produce the vectors that allow us to put CAR T cells, to put the chimeric antigen receptors, the CARs, into T cells. The clinical testing laboratory is equipped to support in-house manufacture of biological therapeutics. We have clinical grade molecular and cellular assays to characterize our products and to perform release testing. Every time we make a product for a patient, this is like a one-off drug, and we have to test that drug to make sure that it fits all of the requirements before we can release it to the patient. And we have 2 ISO Class 7 clean rooms. ISO Class 7 is a standard for the air quality and other characteristics of the rooms in which we manufacture our therapeutics. One of the characteristics of an ISO Class 7 clean room is that all the air that enters into it is sterile filtered before it comes in. So these rooms are designed for the manufacture of cellular therapeutics, oncolytic virus, and viral vectors. It conforms to the FDA mandated specifications for the manufacture of therapeutic products for human use. And so in this way, we are actually conforming to standards that are developed for pharmaceutical companies who produce large batches of drugs. But in this case, where our batch is a single drug, a single dose of drug for a single patient. And it supports our initiative for investigator-initiated phase 1 trials of in-house manufactured immunotherapeutic products. The research laboratory is fully equipped for immunologic monitoring, for cell culture, for assay development, and for cellular therapeutic development. And we have several techniques that we're skilled in and that we're very proud of. Flow cytometry can make multiple measurements on very large numbers of cells one at a time. Dr. Lister showed you examples where we can tell whether a cell has been successfully transfected with the chimeric antigen receptor by flow cytometry. We use it for that and for many other things. We have a multi-analyte bead-based ELISA called Luminex. And what this is capable of doing is measuring as many as 50 different analytes at one time in a very, very small sample. And the kind of analytes that we're measuring are the proteins that are made by the immune system, both when an immune response is successful or when an immune response is unsuccessful. Also, we can measure the kinds of factors that tumors make to suppress the immune response. And we can do this all in a very, very small sample and all in a single sample. And in addition to that, we have facilities for live cell imaging. This is a type of microscopy in which we can make movies of cells in action. And it helps us understand how immune cells interact with tumor cells and how tumor cells can prevent immune cells from killing, or what we can do to overcome this kind of suppression. So the first thing I'm going to talk about, we call them fast TILs. And as I said, they're called fast TILs because we can make them in a relatively short span of time. Now, the first target that we're attacking are cancers that metastasize to the pleura, the space between the ribs and the lungs. And what you can see is an X-ray here. This opacity is fluid that's accumulated in the lungs. This is what the fluid looks like when it's drained, and patients are drained therapeutically because it's very, very uncomfortable to have this kind of fluid pressing against your lung and compressing it. It gives you shortness of breath. And so patients drain several times a week, and often very large amounts, a liter or more. When you look under the microscope, what you see in the pleural effusion are nests of tumor cells. These very, very small dots, these are all immune cells, and you can see how numerous they are. Now, this is different from a solid tumor where the lymphocytes are relatively rare. So unlike solid tumors, tens of millions of potentially tumoricidal T cells home to the cavity cancers where they're silenced by a suppressive environment. And this is the key thing. All of these immune cells are here, and many of them can specifically recognize the tumor cell, but the tumor has the capability of telling these cells to stand down. So one of the most important things to make a cellular therapeutic successful is to change the environment. And right now, Dr. Patrick Wagner is conducting a trial of a drug called tocilizumab, which is an antibody drug that's directed against the interleukin-6 receptor and to neutralize the immune suppression in the pleura and peritoneum. So we envision that when we are able to give these types of cellular therapies, we will give them to a pleural environment that is conditioned. And so fast TILer is a strategy designed to treat cavitary malignancies, both the pleura, as I show here, but also the peritoneum, the belly, by activating and expanding pleural T cells, just like Dr. Samhouri showed you for conventional TIL cells. And we have recently received funding from the Department of Defense to do this, $1.2 million over 4 years, which is enough to treat the first 10 patients. So we said it's rapid. The effusion, draining the effusion to having a drug product to give the patient takes 8 days. And as you saw, it takes a month or so for conventional TILs. That's because we're starting with so many more cells. Local delivery places fast TIL in direct contact with the tumor. Normally, TIL are given into the bloodstream and they have to find the tumor. But because the tumor is in the pleural cavity, we're going to give the cells directly into the pleura through the same catheter that we use to drain the patient. We're also giving interleukin-2, but instead of giving it systemically, where it causes the kind of side effects that Dr. Samhouri described, we're going to give it locally, where most of it will remain in the pleural cavity. And we hypothesize that the adverse symptoms will be much less with this route of administration. So many patients who have pleural effusions have metastases elsewhere, but activated immune cells can migrate out of the pleura into the circulation, where we are hoping that they will attack tumors outside of the pleura as well. Another aspect that I think is very important is that we're making use of the existing CAR-T manufacturing resources at West Penn Hospital. So the same kind of bioreactor, which you can see in this cartoon, that we are using for CAR-T cells, will also be used to make these fast TILs. This process is also extremely cost-effective compared to commercial products. And this is where I put in a plug for in-house manufacturing. It's so important that in addition to our collaborations with industry, we maintain the ability to develop our own therapeutics, because it costs us less than $40,000 to manufacture a product from beginning to end, compared to close to $500,000 for the commercial products that are now on the market. And another plus is that commercial products always come cryopreserved. That is, they're frozen at very, very low temperature, and they're thawed prior to use in the patient. Because we're making them at home, we can give them as soon as they're ready, and we don't have to freeze them and thaw them. And we have some very nice preliminary data in Dr. Lister's in-house CAR-T study that these cells are more effective and work sooner when they are given fresh. Oncolytic viruses have been the research focus of Dr. Bartlett, our cancer center director, for more than a decade. These are engineered vaccinia virus constructs that preferentially infect cancer cells and direct them to secrete factors that provoke an anti-tumor response. So the vaccinia virus, you know, as the smallpox vaccine, but this vaccine has been genetically modified in such a way that rapidly proliferating cells, such as tumor cells, are preferentially infected. And they may be combined with TIL strategies to develop better cellular therapeutics. And that's exactly what we have in the pipeline. In this cartoon, you can see that the process occurs in two stages. First, the tumor is infected with the virus. So this will actually be injected directly into the tumor, and the virus proliferates inside the tumor cells. It kills some, but not all of the tumor cells, but it sets up an environment that counters the tumor-mediated immunosuppression in the environment. So in a way, it's releasing tumor antigens so that the immune system can see them, and it's also reducing immunosuppression. And this is when we come in and we take the tumor cell, we take what's left of the tumors and make conventional TIL cells, as Dr. Samhouri described. And these can either be made conventionally, or we can actually encourage particular types of cells that recognize particular mutations in the patients to grow. And these are infused back into the patient. So when we look at the future of cellular therapy at AHN, we're expanding the cellular therapy program to the new biological therapeutic laboratories at West Penn Hospital. We are looking at immunological priming of tumor with oncolytic virus prior to in-house manufacture and administration. We are designing new retroviral vectors to make new and better CAR-T cells. We are looking at second generation in-house manufactured CAR-T cells, intracranial CAR-T cells for CNS lymphoma. That is an effort that's being pioneered by Dr. Samhouri. I told you about the intracavitary fast T cells with pleural and peritoneal conditioning, and second generation fast TILs selected for better reactivity to tumor antigens. These are all in the pipeline and all will be done in-house. So most importantly, I want to thank our patients They graciously consent to participate in our research, many without any expectation of personal benefit. And they're our most essential partners in this endeavor. And you can see this fortune cookie. We actually got this fortune. Hope is the most precious treasure to a person. And research really is equivalent to hope. We are focused on taking research findings from bench to bedside to community and to optimizing the balance between investigator-initiated early phase clinical trials and products that we manufacture ourselves in our own hospital and our collaboration with industry. And I think by optimizing this balance, we will provide the best treatments for our patients. And I'd also like to recognize the Western Pennsylvania Hospital Foundation and Pittsburgh Foundation, who are responsible for building out the laboratory and funding our in-house CAR T-cell trials, the Department of Defense, who will be funding our FAST-TIL program, A Glimmer of Hope, and the Metaviver Foundation. Glimmer of Hope is a local foundation, and they've been very, very active in supporting research in treatments for metastatic breast cancer. And with that, we'll end and take questions.
Hey, thank you, Dr. Donnenberg. Thanks to all of our speakers. That was very, very informative. We have been answering some questions along the way. I think there is one question that I see here from one of the participants. Is there any form of CAR T cell therapy for breast cancer, specifically hormone positive breast cancer is a question to the panel, to the group.
I can take that one probably, Leslie. So for breast cancer, CAR T is in the very early phase. We don't have any CAR T available for breast cancer, but TIL therapy and TCR therapy, there are clinical trials available for breast cancer. Although the second part of the question is it's usually for triple negative breast cancer because there are many lines of therapy for hormone positive breast cancer and the unmet need in triple negative. So the majority of cell therapy trials are for triple negative breast cancer, not for hormone positive, but reach out, please, to confirm that information. I'll be happy to assist.
Okay, thank you. And with that, I don't see any additional questions in the chat. As I mentioned, some questions have been answered along the way. It is now 6:58, so I think we'll go ahead. Oh, wait, there's another message at the bottom. Let me just check this one here. Oh, how long do T cells last once harvested?
So if I understand the question correctly, so if we harvest the T cells from the patients, how long do they last or live before we can give them to the patient? If that was the question, we have data for in TIL therapy specifically that they can last frozen for up to 1 to 2 years. Of course, we don't know if there is if they can last more because we haven't reached there yet, but 1 to 2 years is the answer to this question.
Okay, a few more questions did pop into the question and answer session. So can any of these therapies be used for CLL is a question from one of the folks.
Yeah, I can take that one. In fact, there's a recently approved product for treatment of patients who are refractory to the conventional forms of therapy for CLL, and our in-house trial, in fact, lets patients participate if they have CLL that's refractory to at least 2 lines of therapy.
Thank you. Another question is, are any of these treatments available for low-grade early stage prostate cancer under study? Any use in prostate cancer?
Nothing available in prostate cancer at this point, and usually if there is any clinical trial for any cancer type, it tends to be for metastatic or advanced stage, not for early stage.
Okay, and is there an age limit for treatment?
Usually not for patients more than 75 to 80-year-old.
Okay, and the last question that I'll take, is the Department of, they're asking if any of your research is funded by the Department of Defense in these scenarios. I don't know that.
Yeah, I can take that one.
Okay.
Yes. The Fast Till program is funded by the Department of Defense, and they have also funded a good deal of the preliminary research that allowed us to do the preclinical work to develop the product.
Okay, interesting. Okay. Well, with that, I think we'll close to questions. We're a minute after 7, and I don't see any additional questions popping into the box at this time. So we would like to thank all of you for attending our Ahead of Cancer lecture this evening. Please join us at our monthly Ahead of Cancer lecture series as we focus on cancer screening, early detection, treatment, and survivorship. As a reminder, tonight's lecture was recorded. If you're interested in rewatching tonight's lecture, check back on the AHN YouTube channel, which is identified here on this slide, for a full recording in about a week or so. This video, as well as other past lectures, will be posted under the Ahead of Cancer playlist. In addition, if you would like to schedule an appointment or have questions regarding a potential cancer concern, please call the Hope line at 412-578-4673 to schedule an appointment. Thank you for the great presentation from our speakers, and we appreciate all who attended the lecture this evening, and have a wonderful evening. You'll also be receiving an email with the Head of Cancer, with the link to the YouTube channel as well. Thank you.